Well, thank you. It's a pleasure to be here. I always feel as if I can only disappoint after an introduction like that. Um, but I think that there's a growing field of the interface between the immune system and the brain, and it's a very exciting one. And what I was asked to talk about today was maternal immune activation and autism spectrum disorder. And I'm really going to divide the talk into a very short part on the innate immune response because Dr. Brown will be speaking more on that, and then talk more about antibodies. So there are good data from epidemiologic studies and some very good animal models to suggest that viral infections and constituents of the virus and cytokines made as a consequence of those constituents by the mother can alter fetal brain development and result in a set of symptoms that are mouse symptoms that are thought to have some analogy to symptoms of autism spectrum disorder. And the mechanisms for this are still being explored. They have to do with altering the placenta or the cytokines from the mother directly affecting the developing brain. And as I mentioned, there are clinical epidemiologic studies that suggest this mechanism for autism also. And this is just to show that the activated immune system from the mother can alter immune cells within the placenta, or some of the molecules can go directly cross the placenta and into the fetal circulation and lead to alterations in brain function. And exactly what kinds of immunologic activation, exactly what panoply of immune mediators might be involved in this, and how they act on developing brain tissue still requires further work. What I'm going to talk about primarily are antibodies and how maternal antibodies might affect fetal brain development. And I'm really going to divide this section into three parts, and first tell you how we came to be interested in this through studies of individuals with lupus, and then talk a little bit about the development of the blood-brain barrier, which is what normally protects us from our antibodies penetrating our brain tissue, and then talk about what we've learned so far about maternal antibodies and autism spectrum disorder. Okay. So it's been known for some time that individuals with an autoimmune disease called systemic lupus, and 90% of those individuals are women, and women usually get the disease in their 20s or 30s. About 20% of cases are actually pediatric. And what we've learned is that there are neuropsychiatric syndromes that are associated with lupus. And these are, as you can see in studies from around the world, quite common, from a low of about 40% to a high of 95%. And that the three most common symptoms are cognitive dysfunction, headache, and mood disorder. And I'm going to tell you a little bit about antibodies we became interested in for their potential in explaining cognitive dysfunction and mood disorder. So what we identified some years ago was a subset of anti-DNA antibodies, and anti-DNA antibodies are the hallmark of lupus. So essentially all patients with lupus have antibodies to nuclear antigens, and the most commonly identified nuclear antigen is double-stranded DNA. And over 70% of lupus patients have antibodies to double-stranded DNA, and they're known to be pathogenic in the kidney and in the skin. And what we identified was a subset of these antibodies that bound to a 5 amino acid sequence that was present in the NR2A and NR2B subunits of the NMDA receptor. And you're neuroscientists, so you know much more than I do about NMDA receptors. They're receptors for the neurotransmitter glutamate. They have 2 obligatory NR1 subunits and 2 of any 4 NR2 subunits, and they've been shown to be critical in learning and memory and in some aspects of mood. And so we were obviously very interested in this because we thought that if these antibodies actually bound NMDA receptors, it would give us really the first insight into pathogenic mechanisms for neuropsychiatric lupus, and indeed for the 2 most common, or 2 of the 3 most common symptoms in neuropsychiatric lupus. So we expressed the extracellular domain of NR2A and NR2B, and we used a monoclonal antibody that is cross-reactive with DNA and this 5 amino acid peptide, and you can see that it bound to both NR2A and NR2B, and a isotype control antibody did not. When this antibody was injected directly into the brain, it caused a loss of nu-N staining, neuronal death. Nu-N is a marker of mature neurons. An isotype control antibody did not. You can see the needle line here and the needle line there. And when we went to hippocampal slice preparations, we could show that the antibody augmented excitatory postsynaptic potentials. So the antibody doesn't cause the activation of the NMDA receptor. You actually need some glutamate or some NMDA receptor ligand to be present. But if it's present, it enhances the activation. And we've actually shown that it prefers the configuration of the receptor, the activated receptor, with an open pore. And so what we believe is happening is that the antibody fixes that configuration for a period of time.
I want to clarify right at the beginning. This same antibody reacts against double-stranded DNA and the peptide?
Yes. So I should just say I can see incredulity rising. Many anti-DNA antibodies have been shown to bind protein antigens. Laminin and alpha-actinin and non-protein antigens like heparan sulfate. And they're just I think that one of the things that my generation has to relearn and teach better is the specificity of T-cell receptors and antibody molecules. And we grew up learning that they're monospecific, and that's not true. And in fact, the introductory comments about Sydenham chorea. There's an antibody to streptococcus that binds to cardiac myosin and dopamine receptors. And the binding to the dopamine receptors, that cross-reactivity is what's responsible for the choreiform movements that you see. So they're not as promiscuous as the pattern recognition receptors of the innate immune system, but cross-reactivity is the name of the game, and I think many immunologists would tell you that that cross-reactivity is part of what's so protective for us. So you make an antibody to virus 1, and some of those antibodies see virus 2, and so when virus 2 comes along, you're not starting off de novo, you actually have some prefab antibodies ready to help you out. And that proteins and nucleic acids might share homology. I'll show you a picture later. I'm not enough of a biochemist to be able to explain that to you in any detail. But one of the hallmarks of many autoimmune diseases and many immunodeficiency diseases is cross-reactivity, polyreactivity, described by Michel Nussensweig. And what he looks for is binding to insulin, to lipopolysaccharide, and to RNA and DNA, and looks for antibodies that are cross-reactive with a protein, a polysaccharide, and a nucleic acid. So this is a not uncommon occurrence.
I want to say that lupus is one of the cause autism, because...
No. There is a paper that says that, but that's not what I'm going to say.
Yeah, because on Philippines is epidemic of lupus and there are no epidemic of autism. I saw some play, even they did play here in Consulate General about lupus, but there was nothing about autism.
So there is a recent paper about children of lupus having autism spectrum disorder, but that's not why I'm talking about lupus. I'm talking about lupus because I think it's a model where we understand some of the molecular mechanisms for antibodies affecting fetal brain development. Okay? So this antibody does cross-react with NMDA receptors and does function to enhance agonists. So it's not a true agonist, but it enhances the effect of agonists. So when we looked in the brains of mice that carry this antibody, because we can actually immunize with the 5 amino acids and have some strains of mice make antibody that is like the lupus antibody, their brains are completely normal. And when we thought about it, we reasoned that that's because we all have a blood-brain barrier. And what the blood-brain barrier does is protect brain tissue from substances in the circulation, including antibodies. And I might even say especially antibodies, because on endothelial cells, vascular endothelial cells throughout the body, there is a receptor for immunoglobulin called FcRn that actually transports immunoglobulin from the bloodstream into tissue. In the brain microvasculature, that receptor has an opposite polarity. It's abluminal, and it actually transports immunoglobulin out of the brain. So we protect our brains from antibodies. But there are known insults to the integrity of the blood-brain barrier, and we looked at 2 of these. One was infection, because lupus patients are treated with immunosuppressive therapy, and so they're prone to infection. And one was epinephrine, because everybody feels stress. And what you can see is, oh, goodness, I'm not very good at this yet. What you can see is that when these mice that were carrying high titers of the antibody were given lipopolysaccharide to mimic infection, antibodies entered the hippocampus, bound hippocampal neurons, caused hippocampal neuron death, and there was an alteration in spatial memory in these mice. And when mice were treated with epinephrine, the hippocampus was just fine. The antibodies entered the amygdala. You're looking at caspase activation in neurons in the amygdala. And these mice had normal memory function, but an abnormal stress response, response in a fear conditioning paradigm. But what one MD PhD student in the lab became interested in is whether these maternal antibodies might actually alter a developing brain, because there is an incredibly consistent literature on the children of mothers and not fathers with lupus. So fathers are much rarer, but they do exist. And having an increased incidence of learning disorders. And so she was wondering whether these antibodies might alter fetal brain development, because there is a time during fetal brain development when there is not a competent blood-brain barrier. And so what she did was immunize female mice. So they made high titers of these antibodies. She then allowed the mice to mate, and then she studied the fetal brains, and she studied the offspring after they were born. And what you see here is that in a brain that's been exposed to or has a mother with high titers of these antibodies, you see an increase in tunnel positive cells, and you see an increase in proliferating cells, and I think quite remarkably, you see ectopic mitosis of neurons. And in mice that are immunized, given adjuvant and immunized just with the backbone that this peptide sits on, there's not much tunnel positivity, and all of the mitotic activity is appropriately in the germinal zone. And what you can see is that the fetal brains had a thinned cortical plate that's quantitated here, and 2 different control immunizations didn't cause any alteration in the thickness of the cortical plate. And to prove that this was antibody mediated, what we did was take a gestating mouse and intravenously give a monoclonal antibody, and this is a mouse monoclonal antibody and a thinned cortical plate quantitated here, and this is a human monoclonal antibody cloned from the peripheral blood of a woman with lupus, leading to a thinned cortical plate, and this is a isotype control antibody, as is this. And this is an anti-DNA antibody, also cloned from peripheral blood of a woman with lupus, but with no cross-reactivity to the NMDA receptor. So not all antibodies that see DNA cross-react with the NMDA receptor. So this showed us that antibody alone, nothing to do with our immunization, was altering the development of the fetal brain. And we, in fact, had 3 cohorts of mice, those that were immunized with an irrelevant, just a polylysine backbone, those that were immunized with peptide on the backbone, and some of those moms made low titers of antibodies and some made high titers of antibody. And we subjected the mice to the equivalent of a mouse APGAR test. It's called negative geotaxis. You put a little pup face down on a slant, and it will turn around and walk to the top of the slant. And what you can see is that the mice with no exposure to this lupus-like antibody and with low exposure to the lupus-like antibody were able to perform this task appropriately well before the mice that were exposed during fetal development in utero to high titer of the antibody were. And when these mice grew up, they were assessed on a large variety of tasks, and they had a normal memory function, and they had a normal stress response. But in two assessments, in just a few assessments, they had abnormal behavior. So if you put a mouse in an environment with two similar objects, you take the mouse out and return it to the environment, but you've now substituted one of the objects. Most mice will spend most of their time exploring the novel object, but the mice that were exposed in utero to high titers of this anti-NMDA receptor antibody do not. And this is a topologic task where you put a mouse in an environment with 4 objects, you change the spatial orientation of 2 of those, and again, most mice will spend most of their time exploring the objects that have been relocated in space, and the mice that were exposed to this lupus-like maternal antibody in utero did not. So you can't call this a learning disability because those are defined by reading and arithmetic skills, and mice, to the best of our ability to assess, don't have those. But these are tasks that require some input from cortical function. That's what happens in the male mice. It's worse for the female mice. You actually have a loss of female fetuses, and Li Wang in the lab was able to show that this is because an earlier expression of NR2A in the brainstem of the female fetuses. So when we looked at a mouse that was deficient in NR2A, there was no loss of female fetuses, essentially the same number of female fetuses as male fetuses were born. And I should say there's one meta-analysis and one study of 900 children of lupus patients, and they both show a striking sex imbalance with more male offspring than female offspring. So that doesn't prove that it's this mechanism, but at least suggests that this mechanism may work. To me, the excitement of finding that antibodies might be responsible for cognitive and behavioral abnormalities in adults and for abnormal brain development in the fetus is that the ability to think about a therapeutic strategy is very, very simple. And so our therapeutic strategy has not been to inactivate the NMDA receptor, though NMDA receptor antagonists will protect, but that doesn't seem a very good therapy. It's to use a decoy antigen. So in adult mice, and here's the amygdala in the triangle. If we have mice that are immunized to make high titers of this lupus antibody and give an epinephrine, you can see dead neurons. But if we give soluble peptide to the mice at the same time, you can see that the antibodies are no longer able to bind their target antigen on the neurons, and they're spared. And again, if we give soluble peptide as opposed to an irrelevant peptide or saline to pregnant mice, there is no thinning of the cortical plate. The cortical plate is of a normal width compared to what you see when mice develop in the presence of this maternal antibody. So this is for the biochemists amongst us. This is the peptide. This is a small molecule orally absorbed that has, in fact, a higher affinity for both a mouse monoclonal antibody with this dual binding to the NMDA receptor and DNA, and the human monoclonal antibody with this dual binding. And we're very interested in fact, going to clinical trials with this for neuroprotection in lupus. And this is just showing you that small molecule, you're looking at tunnel positivity mediated by the mouse monoclonal or the human monoclonal, and this small molecule can protect the neuron from either the mouse or the human antibody. So that study obviously led us to ask about when does the blood-brain barrier develop and how long is the fetus exposed to maternal antibody? Well, it's known that during the first trimester of pregnancy, there's no transport of antibody across the placenta. So we don't need to worry about the first trimester of pregnancy, but we need to worry afterwards. And these are studies that have been done by Cheshire Caval, and some studies I'll show you done in collaboration with colleagues at the Karolinska Institute. So what she did was take pregnant mice at various stages of pregnancy and inject intravenously a infrared labeled antibody. And what you can see is that in the embryonic day 13 mouse, there's antibody throughout the brain, and you can see the disappearance of that. So by E15, there's less. By E17, even less. And this is the mom's own brain. And if you inject these mice with Evans blue dye, which binds to albumin, you can see Evans blue throughout the brain, and by E17, it's in the blood vessels. Albumin isn't crossing across the blood-brain barrier. And if you look at expression of tight junction proteins, which are part of what form the blood-brain barrier, I'm showing you claudin expression, but for any of the tight junction proteins, you can see that they're expressed way earlier than there is, in fact, a competent blood-brain barrier. And the generation of competence of blood-brain barrier really is more seen by MICA32 expression, which disappears, is not present in an adult blood-brain barrier, comes back with brain injury, goes away with repair of blood-brain barrier, and you can see that it sort of mirrors the antibody penetration of the brain. And with Sven Pettersson at Karolinska, we've shown that, in fact, germ-free mice at E16 have much more antibody in their brain than mice raised with gut flora. They look to me like about day E14 mice. And if you look at Evans blue on E16, you see 3 different mice from a germ-free facility, and you can see that they have, in fact, Evans blue albumin throughout the brain, whereas specific pathogen-free mice or conventional facility-grown mice that have gut flora don't have that. And this just shows you, again, immunoglobulin that is not present in the brains of these mice on E16, is present in the brains of germ-free mice. But if you reconstitute the moms with a microbe, in this case Clostridia butyricam, the fetal brain generates a blood-brain barrier. So we're very interested in learning more about what the gut microbiome does to allow you to allow the fetus to make this barrier, and what other what other factors affect barrier development, because we think that it will be important for thinking about antibodies in that maternal antibodies that might affect the fetal brain. So what we think is that antibodies can contribute to cognitive and emotional impairment, and maternal antibodies can contribute to abnormal fetal development. Decoy antigens are a therapeutic strategy, and the blood-brain barrier has to be achieved, barrier function has to be achieved before antibody diversification, and that microbiota are important in this. So this brings us to antibodies and autism spectrum disorder. There have been many studies over the years looking at maternal antibodies given to mice, given to monkeys, and whether the offspring maternal serum or maternal immunoglobulin leads to behavioral changes. These are from mothers with a child with autism, and you can see that these have led to offspring with some features of autism spectrum disorder. Some antibodies, particular antibodies, associate with particular syndromes of frontal lobe enlargement and increased irritability, and mice that naturally have anti-brain antibodies by a naturally occurring mutation have impaired social interactions. There was also a study, I never remember if this is Sweden or Denmark, that showed that children, that mothers with certain autoimmune diseases were at higher risk for having children with autism spectrum disorder. They found arthritis and celiac disease. And so we went to ask the question whether a subset of autism spectrum disorder relates to autoimmunity in the mother. And what we did was look at large numbers of serum samples from mothers of a child with autism spectrum disorder. Over 90% of these or so coming from the Simons Registry. And what you can see is that a higher percentage of these moms had brain-reactive antibodies than of unselected women of childbearing age. And in fact, a smaller percent were completely negative. And I just want to show you our assay is a histologic assay. This is a negative serum. These are positive serum. And then there's some sera that show some pale green without the definition of binding neurons that these have that we called intermediate. Whoops. So when we looked at these moms, there was no difference between the time the child was born and the time the blood was drawn in those that had anti-brain antibodies and those that didn't. There was no difference in the mother's age at the time she gave birth between those who had antibodies and those who didn't. So we looked at another feature of autoimmunity, which is anti-nuclear antibodies. So I began by saying that 95+% of lupus patients have these, but it's 40% of patients with rheumatoid arthritis. It's a large percentage of patients with Sjogren's disease. These are present in many, many autoimmune diseases. And what you can see is that if the mom had brain-reactive antibodies, she was much more likely to have anti-nuclear antibodies than either the moms of an autistic child who didn't have brain-reactive antibodies or unselected women. So this was just a measure that there was other clues towards autoimmunity in these moms. And when we looked for autoimmune disease, we found out that, in fact, these moms were much more likely to have autoimmune disease than the moms of a child with autism spectrum disorder who did not have brain-reactive antibodies. So one of the diseases that had come up in the Danish cohort was rheumatoid arthritis. So we looked at women with rheumatoid arthritis and we asked, do they have brain-reactive antibodies? And indeed, the same incidence of women with rheumatoid arthritis as women with a child with autism spectrum disorder had brain-reactive antibodies. So we would speculate that some of the increased risk that one sees in mothers with rheumatoid arthritis for having a child with autism spectrum disorder may come from these antibodies. So I was asked to say how these antibodies might arise, and of course the answer is, I don't know. I think that even in people with autoimmune disease, microbial exposure may often be the trigger for the initial generation of cross-reactive antibodies. And either with your autoimmune diathesis, you have a repertoire that makes more cross-reactive antibodies, or you don't get rid of them appropriately, or they stay around much longer. So what we've been very interested in is once you form the blood-brain barrier, brain-specific antigens may not participate in the negative selection of autoreactive B cells that I'm going to show you in a minute. And so that means that brain-reactive antibodies may arise in the course of protective responses, and presumably these protective antibodies, as in the case of rheumatic fever, might also be neurotoxic if they manage to get into the brain. So this is data from a review article by Michelle Nussensweig that essentially says in immature B cells, 75% of those B cells are autoreactive, and you purge them from your repertoire. So at the time that B cells acquire immunocompetence, it's only around 15 or 20%. And this process of purging the autoreactive B cells has to do with exposure to the antigen that B cell sees. So if brain antigens are sequestered, there would be no purging of those antigens. So we've actually looked at this, looking at phosphocholene, which is a dominant epitope on pneumococcal polysaccharide, the cell wall pneumococcal polysaccharide, and in some of these serotypes, coupled to either BSA or bovine serum albumin alone. And you can see that in a fear conditioning response, so we then gave the mice epinephrine, and in a fear conditioning response, those mice that had the antiphosphocholene antibodies, in fact, froze longer and were hypervigilant. And you can see when we stain for GABAergic neurons in the amygdala that there's a decrease in the number of GABAergic neurons. And if we just give a monoclonal antibody to a mouse, a monoclonal anti-PC antibody, or an irrelevant monoclonal antibody to a mouse, and give epinephrine, we see the same loss of GABAergic neurons. So this is antibody mediated. So I don't know where they come from, but I think that they can come up during responses to normal infections and just persist, or you may have a larger repertoire of antibodies in response to an infection because you may have an alteration in your threshold for negative selection. I'm coming back to autism now. What Lior Brimberg, who's here in the audience, did was devise a methodology where we could pull these brain-reactive B cells out of the peripheral blood of a mom. So they're less than 1%. And so if you just randomly clone antibodies from peripheral blood B cells, you'll spend a lot of time not finding them. But if you can select for these brain-reactive B cells, you can pull out antibodies, and this is just looking at antibody binding to brain. And this is one monoclonal that binds to an antigen that is a hit on GWAS of autism. So it was very exciting to us to look at. This is looking at this antibody binding to KCNAB2 and not binding to KCNAB1. So it has real specificity. These are other antigenic targets that have all been related to abnormal brain development, either in GWAS studies or in knockout studies in mice that Lior has generated monoclonal antibodies to. So what comes next? What we want to do next is continue to identify more of these antibodies, but really to define which are pathogenic, to put these into gestating mice to see which alter fetal brain development, which lead to a behavioral phenotype in the offspring. We want to use these antigens to develop assays to screen for these in pregnant women so we can actually assess whether reactivity to these targets increases your risk for a child with autism. We want to understand whether we can use these to phenotype patients and whether a particular antigenic specificity will lead to a definable phenotype, because if we just look at all moms with brain-reactive antibodies, there's no phenotype that characterizes their children as opposed to all children. And we want to develop prevention strategies. That's really the most important of all. So I just want to end by really highlighting Lior, who has done all of the autism work, and the others are all individuals who've been involved. And Peter Gregerson, who's an immunogeneticist, has worked on this project also. But everybody else has worked on the lupus project or the blood-brain barrier, and Lior has single-handedly done all of the autism work. And I'm not answering questions now. That comes later.
Well, thank you. Thank you, Lorna, for not embarrassing me. It was a great relief. So I'm one step ahead already. So this is a tough act to follow, really, because what you heard was some really fascinating and groundbreaking work that are furthering, I think, our understanding of antibody-mediated processes in autism and bringing that together with neurodevelopment. I have a few things to say based on what I had an idea of what this talk would be, but not being an animal researcher, I can only speculate a bit about what some of the meanings and connections of this might be to what we're finding in our clinical work, and I'll try to do that as I talk. But clearly this provides really important evidence for the hypothesis that maternal anti-brain antibodies contribute to risk of autism in offspring. And Betty alluded to some earlier work done and some ongoing work done on this topic, but I haven't seen anything really this impressive, especially with regard to the monoclonal antibody effects, the effects on the fetus, as well as the very fascinating work indicating that the probiotics and the gut microbiota play in this process. And this is something that Paul Patterson has been really innovating with in his own work on maternal infection and psychiatric outcomes, including autism phenotypes. So this work, I think, dovetails very nicely as well with the work that I've been doing for a number of years on prenatal infectious and immune disturbances in autism and other psychiatric disorders. And what I find particularly interesting is that the production of these autoantibodies may result both from genetic variants, as Betty was alluding to, and also from infections. So this may represent an interesting point of convergence between genetic and environmental factors, which is, I think, just what this field needs, as well as the fields of schizophrenia and other psychiatric disorders. And it may begin to elucidate a potential pathway mechanistically that Betty is more expert in than I am. There are obviously further questions. One question that I raised would be, are these autoantibodies observed during pregnancy, and is that related to autism? Are these related to other obstetric or perinatal events? These first two questions can be addressed potentially in birth cohort investigations that I'll be talking with you more about. The antigenic targets have already been covered. A big question, I think, too, is, and as was alluded to by Betty's presentation, is, are these autoantibodies specific to autism? The data suggests that abnormalities in cognition and other outcomes. It's also notable that lupus is a known cause of psychosis, at least in adults with psychosis. In the adult patients. And then finally, as a clinician, my question really are there correlations of the autoantibodies with clinical features of autism, as well as neurobiological and neurophysiological features? So there have been, as Betty alluded to, a number of associations of autism in epidemiologic studies, including Denmark, but also other countries, with autoimmune disorders, and these seem to be more common in mothers than in fathers. There are also associations of schizophrenia with autoimmune disturbances, and interesting evidence that these autoimmune disorders may interact with infections and increasing risk of schizophrenia. The PANDAS, or pediatric autoimmune neuropsychiatric disorders, a long name, but these are related to streptococcal infection and involve possible molecular mimicry, have been related to OCD, obsessive compulsive disorder. And in complementing Betty's comments, there have been associations of anti-NMDA receptor antibodies and functional psychoses, as well as mania. So our approach to this was to take birth cohort designs as a way of identifying prenatal exposures and autism, and one of the first things that we studied were, or a set of risk factors, were immunologic and infectious agents. A birth cohort, for those of you unfamiliar, this is a bit of a shift here from animal work, but a birth cohort is a group of individuals who are born within a defined period in a defined place. That could be Alameda County, California. That could be the country of Finland. I mentioned those two because this is where we're doing our studies. And this cohort is then followed up for outcomes of interest, in this case, autism. And I'll be talking about other disorders in a minute. The advantage of the birth cohort is it has a number of methodologic advantages. And I'm not going to talk about all of them, but two key advantages to emphasize are, first, that we're able in a cohort study, because it's a longitudinal study, is to do a prospective ascertainment of exposures during pregnancy using prenatal biomarkers. We've used archived maternal serum specimens in our work that were drawn during pregnancy, and individuals are then followed up for autism. Previously collected obstetric and perinatal records have also been used and very valuable. A second point is that this kind of work minimizes the potential for biases that can arise when the cases and controls aren't equivalent on a number of measures, and so we use what's called a nested case control design, which is a case control study within a birth cohort in which the controls represent the source population giving rise to the cases. Because you can get a lot of spurious findings if you use alternative methodologies that are expedient but not really geared towards answering the question that you have in front of you and really getting definitive results. In previous work, this is a highlight of some of our previous work where we've looked in a California birth cohort, we've demonstrated that a number of prenatal infections are associated with schizophrenia. So we found that maternal influenza in the first half of pregnancy was related to a threefold increased risk of schizophrenia, elevated toxoplasma IgG to a 2.6-fold increased risk. These were both determined based on antibody assays of maternal serum drawn during pregnancy and in individuals who were followed up 20 to 30 to 40 years later for schizophrenia. It's not easy to do that. It took a long time. Records-based studies using physician records have also demonstrated associations of other maternal infections and schizophrenia. We've also been very active in studying cytokines in these epidemiologic studies. Cytokines are a family of soluble polypeptides that are stimulated by and mediate the response to a broad array of infections, are critically responsible and important to the immune response, and elevated maternal and neonatal cytokine levels increase vulnerability to developmental brain damage. We examined second trimester cytokine levels in maternal serum in individuals followed up later for schizophrenia and found that the mothers of schizophrenia cases had about a twofold increase in levels of the cytokine interleukin 8 compared to the mothers of controls. We didn't see these associations for other cytokines, and that may be due to reduced statistical power. We also showed that the higher the level of maternal interleukin 8, the greater the increase in ventricular CSF volume or ventricular size, which has been strongly related with schizophrenia. So that was nice validating evidence using a neural phenotype, not just the diagnosis. We've also extended our findings more recently to look at maternal influenza and risk of bipolar disorder with psychotic features. So in this slide, it shows the results using clinical diagnoses of influenza diagnosed during the pregnancies of these individuals followed up later for bipolar with psychotic features. We found a nearly six-fold increased risk of this outcome in mothers who were exposed to clinical influenza compared to those who weren't, and we see a rather marked increase in the prevalence of influenza in the maternal influenza in cases compared to controls. Interestingly, we saw a very similar finding, nearly identical odds ratio, about a 5-fold increased risk when we assayed the maternal serum for influenza antibody. So these actually were ascertained independent of one another, and yet we get very similar findings. So now to move on to autism, there have been a number of studies that have looked at prenatal cytokine disturbances and infection in autism. Increased maternal serum levels of several cytokines have been demonstrated in the early markers of autism study in California. In amniotic fluid in a Danish birth cohort, a number of cytokines were found to be increased. Some of them were the same as those in the serum, especially IL-4 and IL-5. One of the issues that comes up with amniotic fluid, though, is the source of the cytokines is unclear. The contribution of the fetal and maternal compartments is a question. These are really good studies, very innovative. They do have the potential for false positive results because these studies generally look at many cytokines at one time and report all their findings, but they generally don't adjust for multiple comparisons. However, there is some consistency, as I mentioned, in the specific cytokines. In these studies, there is also a lack of information, a high rate of non-participation. Prenatal infection has also been associated with autism. One of the first infection that's been related is rubella. It's a rare exposure, but informative. In those original study by Stella Chess, and I'm very familiar with this because we did a study on schizophrenia in that same cohort, the autism diagnoses weren't obtained by structured interviews, and there was no unexposed comparison group. In schizophrenia, we found a 15-fold increased risk of schizophrenia when we used a structured diagnostic interview. Maternal viral infection has also, in the first trimester, and bacterial infection in the second trimester have been associated in a Danish cohort with autism risk. These studies are based solely on records of hospitalization for maternal infection. That's very interesting, but it doesn't cover most infections that occur in a population-based study. Maternal influenza has also been reported to be borderline significant. This is based on maternal interviews, which may not always be reliable, rather than on clinical records or on serum biomarkers. We got interested in C-reactive protein, in part because it's an established marker of low-grade inflammation from both infectious and non-infectious exposures and is substantially elevated during acute infection. CRP can be induced by a variety of factors other than infection, including chronic inflammation as well as tissue injury, which induces the release of pro-inflammatory cytokines that in turn stimulate the release of C-reactive protein from the liver. We had the great fortune of being able to implement these, address these questions in a large birth cohort in Finland called the Finnish prenatal, which we call the Finnish prenatal study of autism. And we originated this study a few years ago. This is a national birth cohort study based on the Finnish maternity cohort, which consists of 1.5 million pregnancies, representing virtually all pregnant women in Finland from 1983 to the present. This sample contains archived prenatal serum specimens in the first and second trimesters that were stored frozen in a single biobank from all of these pregnancies. We were able to implement the nested case control design, and were able to study all offspring born in Finland from 1987 to 2005. Finland, like a lot of other Scandinavian countries, has these incredible registries that are very comprehensive. So using the registries and doing linkages, we were able to identify nearly all cases of childhood autism in Finland, over 1,100 in the sampling frame we studied, and over 4,000 cases of ASD. So we got to very large sample sizes, which improves statistical power and allows us to do a lot of other interesting analyses. The controls were drawn from the same cohort. They had no ASD or severe profound mental retardation. In this C-reactive protein study, we looked at 677 matched case control pairs from the FIPS-A study. And we used a standard assay method. What this slide shows now are our results. So we broke the sample down into quintiles. These individuals represent mothers who had CRP levels in the highest 20th percentile of the distribution. And we found here about an odds ratio of 1.43, meaning a 43% increased risk of autism in their offspring, so in the mothers in this group. And you can see here the cases being greater than the controls. So that was intriguing. We then looked to see whether the findings may be confounded. We did analyses adjusting for known covariates and found that the results were nearly similar with the unadjusted and adjusted models for highest quintile. We also looked at the highest decile of the distribution, so the highest 10 percentile, as you know. And we found in the adjusted analysis a higher rate, about 83% increased risk of autism. This finding is in molecular psychiatry, will be coming out soon. I'd like to conclude by noting what are, in my view, priorities for future epidemiologic and translational research on immunologic disturbances in autism, and also ways that we may be able to bring these two fields together, which I think is not done significantly enough. One of the most important of these, I think, is gene-environment interplay. And so this work, in my view, what Betty presented and what I'm presenting, could serve potentially as a means for gene identification, given that there are genetic variants that increase susceptibility for autoantibody formation, possibly following infectious insults, as she alluded to. In addition, this work may lead to a strategy for gene-environment interaction. And it's now been shown that a few polymorphisms in MHC in the major histocompatibility complex, which regulates the HLA response and the immune response, have been associated with autism and with schizophrenia. There have been several now good replication studies, genome-wide association studies in schizophrenia. And I suspect that there are some recent data now indicating that this may also be true in autism. So what I believe is essential is the problem, I think, the challenge that we face is that in epidemiologic studies, we typically focus on environmental exposures, and in genetic studies, we focus on genes and not environmental exposures. My view of what's sorely needed is to collect both genetic and prenatal environmental data on cases and controls. And if we're looking at the prenatal environment, ideally, I think we would want to study birth cohorts. And these cohorts exist, and I mentioned one of them. This work also has implications for translational research in terms of probing pathogenic mechanisms, as Betty alluded to. In addition, it would be, I think, very valuable, even though her data appear it seems likely that these antibodies are present during pregnancy. We can actually document that in these birth cohorts, as well as look at their relationship to autism and to infectious insults that we're also measuring in this birth cohort. And another question I think that's important is whether these elevations in cytokines, CRP, and autoantibodies might represent a common pathway by which maternal infections increase risk for autism. With the data that we're collecting on infection and on these antibodies, we hope to be, and on cytokines, we hope to be able to address that question. This has obvious questions as an epidemiologist, has obvious questions for prevention, or implications for prevention, early intervention. And I think it also, as I alluded to in the earlier work, has implications for specificity. The question really is, do these maternal infections and autoantibodies increase risk only for one disorder or only for autism? It doesn't appear to be the case, but it may be that in interaction with particular susceptibility genes that may be unique to each of these disorders may result in or may help determine the final outcome. So that's all I have to say. I'd like to acknowledge my many collaborators in my unit in birth cohort studies, Department of Epidemiology, Kaiser, Finland, CHDS, the birth cohort study in California, and my funding sources. Thanks for your attention.